Welcome to The Explainer. Today, we're diving into one of biology's greatest magic tricks, how every single one of your cells crams a 2-meter-long DNA molecule into a space that's literally thousands of times smaller. It's an amazing story. So here's what we're going to cover. We'll start with a mind-bending packing problem. Then we'll see how nature solves it with some incredible elegance. And finally, we're going to explore what happens when this super intricate system goes wrong. All right, so to really get chromosomes, you first got to wrap your head around the absolutely absurd challenge your cells are up against every single day. Take a look at this number, 2 meters. What do you think that represents? Well, that is the length of the DNA from a single one of your cells if you stretched it all out. 2 meters of genetic code. And this is where things get, well, kind of insane. You have to fit those 2 meters of DNA into a cell nucleus that's only about 10 micrometers wide. To put that into perspective, that's like trying to cram a 20-mile-long thread into a basketball. I mean, how is that even possible? So how on earth does the cell pull this off? Well, it uses this incredible multi-level folding process that is, honestly, both brilliant and beautiful. Okay, it all starts with these little proteins called histones. The best way to think of them is as microscopic spools for our genetic thread. Without these, the DNA would just be a tangled disaster. They're the absolute first line of defense against chaos. So the DNA wraps itself around these histone cores, and what you get is a structure called a nucleosome. This is the basic, fundamental unit of packaging. If you look at it under a powerful microscope, it actually looks like beads on a string. It's this first, really beautiful, repeating pattern in this whole complex origami. Now, you take all of that, you've got the DNA thread, the histone spools, and a few other proteins that help with structure, and the whole shebang together, that's what we call chromatin. This is the actual stuff our chromosomes are made of. So it's not just DNA. It's this incredibly organized, composite material. And here's how that compaction happens, step by step. First, the DNA wraps around the histones to make those nucleosomes, and boom, it's already 7 times shorter. Next, those nucleosomes coil up into a really dense fiber, and that gets you a 50-fold reduction in length. Finally, other proteins come in and pack that fiber into the super compact chromosome, which is so dense you can actually see it with a light microscope during cell division. Okay, so we've got these perfectly packed little genetic suitcases. What's next? Well, the next critical task is figuring out how to distribute them correctly to create our sex cells, our eggs, and our sperm. And this is a hugely important question, right? Because if you get this wrong, the implications are massive. The answer lies in a really special kind of cell division. That process is called meiosis. Now, you've probably heard of mitosis. That's how your body makes identical copies of cells for, growth and repair. Meiosis is a whole different ballgame. Its entire purpose is to cut the chromosome count exactly in half to create sex cells, or what scientists call haploid cells. Think of it like this. Most of your body cells are diploid, meaning they've got the full 46 chromosomes, arranged in 23 pairs. But meiosis creates haploid sex cells, eggs and sperm, with just 23 single chromosomes. And this is just brilliant, because when an egg and sperm combine, the new organism gets the perfect set of 46. But, meiosis is this incredibly precise, high-stakes biological dance, and sometimes someone misses a step, and the consequences can be really serious. That failure, that misstep, has a name. It's called nondisjunction. It's really just a fancy term for when the machinery of cell division makes a mistake and you end up with eggs or sperm that have the wrong number of chromosomes, either one too many or one too few. And when this error happens is a huge deal. If the mistake happens early on, in the first stage, meiosis 1, bam, all 4 of the resulting sex cells will have the wrong chromosome number. But if it happens later, in meiosis 2, only half of them will be affected, which, of course, still has major consequences. So what happens when one of these abnormal gametes is part of fertilization? Well, that can result in certain genetic conditions. You can see a few examples here, like Down syndrome, which is caused by an extra copy of chromosome 21, or Turner syndrome, which comes from a missing X chromosome. Each one of these conditions is tied to a very specific chromosomal error. Okay, so we know these errors can happen, but how do we actually see them? Well, scientists came up with a pretty ingenious tool to get a direct look.
It's called a karyotype. And honestly, the best way to think about it is like a genetic mugshot. Scientists take a cell, they stop it right in the middle of dividing when the chromosomes are all condensed and visible, and then they arrange them into this organized profile. And by arranging all the chromosomes in their matching pairs, from biggest to smallest, doctors can spot any problems pretty much at a glance. They check the total count, is it 46? They look for matched pairs, and they verify the sex chromosomes. Any deviation from the norm just pops right out. All right, for our last stop today, we're going to look at something really fascinating, a specialized case where the body intentionally tinkers with a chromosome to solve a fundamental genetic puzzle. So think about this for a second. Females have 2 X chromosomes, XX. Males have 1, XY. So why don't females produce double the amount of all the gene products from that X chromosome? I mean, that would be a huge dosage problem for the body. Well, the solution is just unbelievably elegant. The body just shuts one of them down. Early on, when a female embryo is developing, each cell randomly and permanently inactivates one of its 2 X chromosomes. This ensures a balanced genetic dose between males and females. And if you want a perfect walking, purring example of this, just look at a calico cat. See, the genes for orange and black fur are on the X chromosome. In female cats, as the embryo develops, each cell randomly shuts down one X. If the cell turns off the X with the orange gene, that cell and all its descendants will make black fur. If it turns off the black X, they'll make orange fur. And the result is this beautiful, random mosaic of color patches. That inactivated X, by the way, condenses into a little structure called a bar body. So, from a 2-meter strand of DNA all the way down to a single chromosome being switched off, the story of our genetic code is just one of immense scale and breathtaking precision. I think the key thing to take away from all this is that how our DNA is packaged and managed is just as critical as the information it holds. It's a great reminder that in biology, the instruction manual and the bookshelf it sits on are equally important.